Hello everyone, my name is Robert Collotti from Queen's University, and the work I'm presenting is part of a manuscript that's in preparation, looking at the role of trade-offs in niche shifts and range limits in novel and changing environments. Under the classic model of local adaptation, we typically have some kind of environmental gradient in which fitness is defined by stabilizing selection within an environment, but divergent selection across environments. If we do a classic reciprocal transplant experiment where we move locally adapted populations and grow them at their home and away sites, we should see that a given genotype or population does better in its home environment than it does when it's grown away. If we compare different genotypes or populations growing in the same environment, then we should see that the local genotype does better than the foreign genotype. If we look at individual alleles, we should typically see this phenomenon of antagonistic pleiotropy, where the local allele does better than the foreign allele in both environments. The problem is that a number of empirical studies over the past 20 years or so don't seem to support these predictions. So these papers cited in the bottom right-hand corner are a couple of meta-analyses. And both studies found when genotypes are grown in the same environment, the local genotype does better than the foreign genotype, but less than half the cases support the prediction that genotypes perform better when they're grown in their home environment than when they're grown away. There are also studies looking at natural selection, and these studies show that stabilizing selection is quite rare in natural populations, and directional selection is quite common. Here's an example of a meta-analysis on flowering time. Directional selection for early flowering is common, with the strength of directional selection changes with latitude. And then a few years ago, we looked at evidence for antagonistic pleiotropy. It's actually very rare. Let me give you an example from purple loosestrife to put some biological context here. So we did a reciprocal transplant experiment running over 4 years. We measured selections on an F2 segregating population, as well as testing local adaptation of the parental genotypes. This is the result, and it's very consistent with what we saw in the meta-analysis. If you compare the local versus foreign genotypes. So here's the northern garden, fitness in the northern garden. You can see the northern genotypes do better than the southern. And in the southern garden, the southern genotype does better. But the home versus away criteria is not supported. In fact, the northern genotype does equally well or even better as you move it south. The reason why we get local adaptation is it's the southern genotype that gets hammered when we move it north. If we look at natural selection on the F2s, we find that selection is mostly directional, again, in line with those meta-analysis studies. You can see that selection favors earlier flowering, but the slope of this increases as we move north. So how do we get local adaptation under directional selection? We think that there's a developmental constraint on flowering time that limits the response to natural selection. So here on the right, I've mapped the same fitness surfaces and a principal component axis defining a trade-off between flowering time and size at flowering. Another way to think about this is if we just take a cross-section of this first principal component axis and look at the fitness function in the southern environment, what happens as we ratchet down selection? We get a shift in the optimum, and we get a ratcheting down of the maximum fitness that a population can obtain. And I just want to promote Yihan Wu's talk. As a former master's student in my lab, she'll be presenting some evidence that latitudinal declines evolved repeatedly and rapidly with the rate of evolution slows after about 100 years. So returning to our classic model of local adaptation, to make it more consistent with the data that we observe in purple loosestrife and in those meta-analyses, rather than thinking about an environmental gradient with different optima for different genotypes having the same optima but different constraints. This genotype might be like our southern populations of purple loosestrife, which get much bigger in the long growing season, but as growing season gets shorter, fitness rapidly declines. In contrast, the northern genotypes maintain fitness across a broader range of that environmental gradient, but suffer a cost of reduced fitness in that southern environment. Similarly, we would make slightly different predictions where we still have the local versus foreign. The local genotype does better, but it's not a home versus away advantage. It's a high quality versus low quality. They both do better in that higher quality southern growing environment. Here's another example of the model with another former student, Richie Honor. And the idea here is just that when species are introduced, they leave behind a lot of their natural enemies, especially specialist natural enemies. And we're interested in how this will affect population growth rates and the rate of expansion of invasive species. This was recently published as a book chapter, and the biotic axis, you can think of this as a gradient from low enemy abundance to high enemy abundance. And then the I and N is just the range of enemy abundance in the native and introduced range. And then we have these different reaction norms for different genotypes, with the blue genotype representing a resistant genotype that maintains fitness across a wider range of biotic activity, but suffers a cost of lower fitness when enemies are rare or absent. We've started to explore this as interactions along an abiotic gradient. So what we've done here is just to take the Lotka-Volterra model of population growth. We define the niche where population growth at low density is greater than one individual per individual. We also assume that soft selection is minimal, and then we simply scale fitness using a Gaussian function. So here's one example for a biotic gradient, but the orange genotype always does better than the blue along the biotic gradient, but it also does worse along the abiotic gradient. So it's a trade-off between axes. And here's one example of a prediction we can get out of this model for a reciprocal transplant experiment in 4 environments. So we have 3 common garden locations within the range and 1 common garden outside of the range. And each common garden represents a different combination of the biotic gradient and the abiotic gradient. This might be like a low enemy southern environment, low enemy northern environment, high enemy southern environment, and high enemy northern environment. And if we compare the predicted fitness in each pair of these 4 environments, we can see that for a given allele, we predict mostly neutrality or conditional neutrality. Only if we compare these 2 environments within the range will we see evidence of antagonistic pleiotropy. We can also think about this in terms of enemy release. And here we're looking at a shift from a high enemy environment to a low enemy environment. And then this panel is showing niche size for the different genotypes. As we reduce the number of enemies, there's an overall increase in the niche along the abiotic axis, but we get different predictions for which genotypes are responsible for that shift. And it completely comes down to the structure of the trade-offs within and between these 2 different axes. Here we're looking at 4 different columns representing 4 different trade-off structures. And now we're looking at an optimum, at a midpoint along the abiotic gradient, and imagining covariance between our biotic and abiotic gradient. And the inspiration here was thinking about latitudinal gradients or altitudinal gradients, where you often have covariance between, for example, environmental productivity and herbivory. And we have this example of enemy release where we're really just shifting down the biotic axis and making this covariance weaker. And here we're showing the expected population growth rates under the original scenario and the enemy release scenario. And again, we get differences in the representation of genotypes and the size of the range expansion, depending on the structure of the trade-offs in the different parameters of the fitness functions. We also can look at the evolutionary rate here, which is just the variance in fitness among the genotypes at different locations along the range under different scenarios. And we get shifts that favor one genotype over the other in these different environments. Okay, so just to summarize, the classic model of local adaptation makes a number of predictions that are not well supported by the empirical literature. So we introduce a trade-off model, and these models are consistent with data that we see in purple loosestrife and in the meta-analyses. So as I mentioned, that paper will be submitted soon to the Philosophical Transactions of the Royal Society. And if this general area of local adaptation and environmental change is something that interests you and you're looking for graduate or postdoc opportunities, I would love to hear from you. Thank you.